Welcome back, everyone. Ready to dive into something pretty complex.
I'm ready.
All right, then let's talk blood groups.
Sounds good.
But not just the ABOs that everyone knows.
Right.
We're talking about the MNS system.
Oh, wow.
It's got over 50 different antigens.
Really?
Yeah, so it gets pretty complicated.
I can imagine.
And it's really important for making sure that blood transfusions are safe.
Absolutely, yeah.
And get this, we're going to learn all about it without doing any experiments.
How's that?
We're going to be looking at a PhD thesis.
Oh, very cool.
And this thesis actually combines real lab work with computer modeling.
That is a fascinating approach.
So using these computer models, we can really get a lot of insight into how things work at a molecular level.
It's incredible what we can do with computers these days.
Yeah, so this thesis takes us through the researcher's journey
Okay.
to create monoclonal antibodies,
Monoclonal antibody.
or MABs, for the MNS blood group system.
Okay.
And these MABs, they're like highly specialized keys.
So
each one is designed to identify a very specific blood type.
just like a key fitting a specific lock.
Exactly.
Got it.
And they're essential for making sure that the right donor blood goes to the right recipient.
Of course, that makes sense.
Yeah. So the researcher starts by trying to create these MABs
Okay.
using small pieces of the MNS proteins.
So they're using like fragments?
Exactly. They're called peptides.
Okay.
It's a pretty standard approach,
But,
but here's where it gets interesting.
okay, listening.
The antibodies bound perfectly to those isolated peptides,
Okay.
but then they completely failed to recognize the same targets on real red blood cells.
Oh, so they made the keys, but they didn't work on the actual door.
Exactly. It's like they had a blueprint that wasn't quite accurate.
So what happened? How did they figure out what was going wrong?
Well, there was a big clue
Okay.
from a technique called Western blotting.
Right.
And the antibodies showed this really faint,
Okay.
almost ghostly binding to red blood cell ghosts.
Red blood cell ghosts. What are those?
They're basically just empty red blood cell membranes.
Oh, so like the shell of the cell.
Exactly.
Interesting.
And that was a big hint
Yeah.
that the structure of those MNS proteins on real red blood cells
Okay.
was much more complicated than just the simple peptides that they were using in the lab.
So like the key was the right shape, but the door was more intricate than they thought.
You got it. And this is where computer modeling comes in.
Computer modeling.
Yeah. They needed to see the full three-dimensional shape of these proteins.
Right. To understand why those antibodies weren't binding.
Exactly. And they focused on three key proteins.
Okay.
Glycophorin A,
Okay.
glycophorin B,
Got it.
and a hybrid version called GP-MER.
So these glycophorins are the ones that carry those MNS antigens on the surface of the red blood cell.
Yes, and they are far from simple.
Oh, I bet.
They're covered in tons of sugar molecules.
Really?
It's called glycosylation.
Okay.
So imagine trying to study a delicate sculpture that's covered in layers of frosting.
Oh, wow.
Traditional methods really struggle to see past that sugary coating,
Yeah, I can see how that would be a problem.
but computer models can see right through it.
Wow, that's amazing.
That's like having X-ray vision for proteins.
I like that.
So what did they find?
Yeah, what did they see?
Well, they discovered a really important structural element.
Okay.
All three of those proteins
Yeah.
showed a recurring fold.
A fold.
Yeah, it's called a beta hairpin.
A beta hairpin.
Okay, and this fold was especially prominent in a region called the exon 3 to 4 junction.
All right, why is that important?
Well, what's fascinating
Yeah.
is that this region also happens to be where many of those MNS antigens are located.
Oh, so we're talking about a specific fold
Right, the beta hairpin.
in this protein structure.
Exactly, and it's crucial,
Yeah.
both for the protein's overall shape
Okay.
and for how those antibodies might recognize it.
So it's like the keyhole itself might be part of that little beta hairpin fold.
Precisely.
Yeah.
So the problem wasn't just the sugar coating.
Right.
It was that those simple peptides that they were using to create the antibodies
Yeah.
couldn't form that crucial bend, that beta hairpin.
They were missing a vital piece of the puzzle.
Exactly.
Wow.
So they knew what they needed,
Right, the beta hairpin.
but they couldn't get those peptides to cooperate and fold themselves in the right way.
That must have been frustrating for the researcher.
Oh, I'm sure it was.
So what did they do next?
Well, faced with those challenges, they took a really innovative approach.
Really?
They shifted their focus
Okay.
from mimicking natural structures
Right.
to actually modifying the antibody itself.
Oh, wow. So they're redesigning the antibody.
Exactly.
How is that even possible?
It's not like they can pick up a molecular scalpel and start carving.
Yeah, That's what I was thinking.
Right. But remember that CDR3 region we talked about?
The CDR3 region,
The grabbing hand of the antibody.
the part that actually latches onto the antigen.
You got it. Well, the researcher explored ways to tweak that CDR3 region
Okay.
so that it could still bind to the M&S antigens, even if
Even if what?
the target proteins weren't folded into that ideal beta hairpin shape.
So they were trying to make that grabbing hand more adaptable?
Exactly. And to do that,
Yeah.
they focused on the properties of beta sheets.
Beta sheets.
You know, those stable structures formed by interactions between amino acids.
Right.
Well, it turns out that beta sheets can form
Yeah.
not just within a single protein,
Okay.
but also between different proteins.
Oh, so like a bridge connecting them.
Exactly. And that's exactly what the researcher was aiming for.
So they wanted to create antibodies that could form a beta sheet connection
Yes.
with the M&S proteins, even if those proteins weren't folding into that ideal beta hairpin shape.
You got it.
But how do you force a beta sheet to form between two different proteins?
It's definitely a challenge.
It sounds like it.
But the researcher had a pretty clever strategy.
Okay, tell me more.
They started by analyzing the amino acid sequences of the M&S proteins,
Okay.
specifically those regions that are associated with the J and U antigens.
Right, the ones they were targeting.
Exactly. It's like reading the instruction manual for those proteins.
So they were trying to understand how those proteins were supposed to be shaped.
Precisely. And based on that analysis,
Yeah.
they were able to design short peptide sequences
Okay.
that acted as mirror images.
Mirror images.
Yeah, the target regions on the M&S protein.
Oh, that's fascinating.
So imagine folding a strip of paper in half.
Okay.
The two sides are mirror images.
Right, they fit together perfectly.
The researcher did something similar with amino acids.
Wow.
They created these short peptides that,
Yeah.
if folded into a beta sheet,
Okay.
would perfectly match and bind to the target regions on those M&S proteins.
So they were creating custom-made beta sheet magnets
You got it.
to stick onto those M&S proteins.
Exactly. Even if the proteins themselves weren't perfectly shaped.
Brilliant. So what happened next?
Well, the next step was to attach those custom peptides
Okay.
to the CDR3 region of the antibodies,
All right.
effectively giving those antibodies beta sheet grabbing hands.
Oh, I love that analogy.
Specifically designed to latch onto those J and U antigens.
Okay, so they redesigned those antibodies with these special beta sheet magnets
Right.
attached to their CDR3 regions.
Exactly. But did it work?
That's the million-dollar question, isn't it?
Yeah.
Could these modified antibodies actually bind to those M&S proteins on real red blood cells?
I'm on the edge of my seat.
Well, we're going to have to wait to find out.
Oh, come on.
You're kidding?
I know it's a bit of a cliffhanger.
It is.
But this particular thesis focuses on the design and development of this new strategy.
Okay.
The actual testing
Right.
is left for future research.
I guess that makes sense, but I'm dying to know if it worked.
I understand. But even without knowing the definitive outcome,
Yeah.
the implications of this research are huge.
I can see that.
There's a whole new way of thinking about antibody development.
Absolutely.
And it has the potential to overcome a lot of those challenges that
Yeah.
we faced with traditional methods.
For sure.
So even if this specific attempt wasn't successful,
Okay.
the fact that they even came up with this idea and figured out how to do it
Right.
is groundbreaking.
It really is.
And it highlights how computer modeling is changing the game
You got it.
in biology and medicine.
We're not just observing nature anymore.
Yeah.
We're designing and engineering solutions.
Exactly, at the molecular level.
It's like we've gone from reading the book of life
Yeah,
to actually having the power to edit it.
and this is just the beginning.
I'm excited to see where it goes from here.
Me too.
Hey.
I'm curious, though, did the thesis explore any other applications of this technology?
It did, actually.
Oh, wait.
Remember those hybrid glycophorins we talked about?
The ones that are a mix of GPA and GPB.
Exactly.
Yeah.
The researcher also used computer modeling to study those hybrids.
Oh, wow.
And they found some pretty interesting things about how they might affect antigen presentation.
Okay, now I'm really intrigued.
Yeah, it's pretty cool.
Tell me more about these hybrid glycophorins.
So they focused on one hybrid in particular
Okay.
called GP-MER,
GPMR.
and they created a computer model of this protein,
Okay.
and then they simulated how it would interact with antibodies.
Got it. And what did they find?
Well, they found that the presence of this hybrid protein
Okay.
actually seemed to mask certain antigens.
Mask certain antigens.
Yeah, making them less visible to those antibodies.
Wait, so these hybrid proteins can actually hide antigens?
That's what it seems like.
It's like having a secret door that blends in with the wall,
Exactly.
making it invisible to anyone who doesn't know it's there.
Right.
But if certain antigens are hidden, wouldn't that make blood typing and transfusions even more difficult?
It's definitely something to consider.
Yeah,
This finding really highlights the complexity of the MNS blood group system,
for sure.
and it emphasizes how much we still have to learn. Absolutely.
But research like this,
Uh-huh.
using computer modeling to understand these intricate interactions,
Right.
is paving the way for more accurate and personalized blood typing in the future.
It's amazing how much we can learn from these computer models.
It really is.
It's like having a molecular microscope
Yeah,
that allows us to see the hidden world of proteins and cells.
I love that analogy.
But I have to admit my brain is starting to feel a bit overloaded.
I know, it's a lot to take in.
It is.
Maybe we should take a moment to let everything sink in.
That sounds like a great idea.
Okay.
We'll be back in a moment to wrap up our deep dive into the MNS blood group system and explore some of the broader implications of this research.
Sounds good. It's a lot to process for sure,
Definitely.
but it's so cool.
It is.
So much going on at the molecular level.
All right, well, feeling refreshed.
Yes, definitely ready to keep going.
All right, so we were talking about how researchers are using computer models
Right.
to actually design more effective antibodies.
It's pretty amazing stuff.
Ugh.
But before we go any further,
Okay,
I want to take a step back
great.
and consider the bigger picture.
Sounds good.
Why should someone who's not a blood specialist care about this research?
That's a great question.
What's the takeaway for the average listener?
Well, on a practical level, this research could lead to more accurate blood typing
Okay.
and safer transfusions.
Especially with the discovery of those hybrid glycophorins
Right.
that can mask certain antigens.
Exactly. We don't want any hidden antigens causing problems during transfusions.
Absolutely not.
But beyond blood typing,
Yeah.
this research represents a major shift in how we think about developing antibodies.
Okay.
This idea of directly modifying antibodies, using techniques like those beta sheet magnets,
Right.
opens up so many possibilities for treating all kinds of diseases.
It's like we're not just relying on nature's blueprints anymore.
Exactly.
We're becoming molecular architects,
I like that.
designing and building our own solutions.
It's really exciting.
What other potential applications are there for this technology?
Oh, the possibilities are huge.
Like what?
Well, think about autoimmune diseases.
Where the immune system attacks the body's own cells.
Exactly. What if we could design antibodies to specifically target those rogue immune cells?
And neutralize them.
Yeah.
Or what about cancer?
Oh, that's a great one.
Could we design antibodies that are like guided missiles,
I love that analogy.
seeking out and destroying cancer cells
Right?
while leaving the healthy cells alone?
That's the goal.
It's amazing to think about.
Really is.
This research is just one piece of a much larger puzzle.
For sure.
Scientists all over the world are using computer modeling to study all sorts of things.
Absolutely.
From protein folding to drug interactions
Yeah.
to the evolution of viruses.
It's incredible.
We're entering this era where our understanding of biology is becoming so precise.
And our ability to manipulate it is growing so rapidly.
It's thrilling,
It is.
but also a little daunting to think about the possibilities.
And the responsibility that comes with this level of knowledge.
Exactly.
We have to use this knowledge wisely and ethically.
Absolutely. But I'm optimistic that this new era of biological understanding will lead to incredible advances
I agree.
in medicine and human health.
For sure. Who knows what diseases we'll be able to cure?
What lifespans we might be able to extend.
What mysteries of life will unlock?
The potential is truly limitless.
I love that.
Well, this deep dive has certainly left me feeling optimistic
Me too.
about the future of scientific discovery.
It's amazing to think that even a topic like the MNS blood group system
Right.
can lead to such profound insights into the future of medicine and human health.
It really shows how every discovery, no matter how small, can have a huge impact.
That's the beauty of science.
We've given our listeners a lot to think about today.
We have.
But before we wrap things up,
Okay.
is there one final thought or provocative question you'd like to leave our audience with?
Here's something to ponder. We've talked about computer models as tools for understanding existing biological systems.
Right.
But what if we could use them to design entirely new biological systems?
Oh,
Ones that don't even exist in nature.
now that's a thought-provoking question.
I know, right?
We might need a whole other deep dive to explore that one.
We probably would.
But for now, I want to thank you for joining us on this fascinating journey into the world of blood groups and beyond.
It's been a pleasure.
And for those of you who want to learn more, we'll have links to additional resources in the show notes.
Be sure to check those out.
And remember, this deep dive has only scratched the surface.
There's so much more to explore.
Keep asking questions.
Keep learning.
And keep diving deep into that vast ocean of knowledge that awaits us.
Absolutely.
Until next time, stay curious. All right, so much to think about, right?
Yeah, those hybrid glycophorins really threw me for a loop.
It's incredible how complex this system is.
It really is.
We've learned so much about the MNS blood group system.
And those sneaky hybrid glycophorins.
And the incredible ways that researchers are using computer models
To design those more effective antibodies.
It's really revolutionizing our understanding of biology.
Especially at the molecular level.
But before we wrap up,
Yeah.
I want to step back for a second.
Okay.
And think about the big picture.
All right.
Why should someone who's not a blood specialist care about this research?
That's a great question.
What's the takeaway for the average listener?
Well, on a practical level, this research could lead to more accurate blood typing,
Okay.
safer transfusions,
Makes sense.
especially with those hybrid glycophorins that we talked about.
Right? They can mask certain antigens.
Exactly. We don't want any surprises during a transfusion.
Absolutely not.
But beyond just blood typing,
Yeah.
this research represents a real paradigm shift
Okay.
in how we think about developing antibodies.
I see what you mean.
This idea of actually modifying the antibodies themselves,
Yeah.
using techniques like those beta sheet magnets,
Yeah.
it opens up a whole world of possibilities.
For treating all sorts of diseases.
Absolutely.
It's almost like we're not just relying on nature's blueprints anymore.
Yeah.
We're becoming molecular architects,
I love that analogy.
designing and building our own solutions.
It's really exciting.
What other potential applications can you think of for this technology?
Oh, the possibilities are endless, really.
Like what? Give me some examples.
Well, think about autoimmune disease.
Yeah, where the immune system attacks the body's own cells.
Exactly. What if we could design antibodies that could specifically target those rogue immune cells,
Like just neutralize them.
right?
Or how about cancer?
Oh, that's a big one.
Could we design antibodies that are basically like guided missiles,
I love that.
seeking out and destroying those cancer cells
Right.
while leaving the healthy cells unharmed?
The dream, right?
It's incredible to think about.
It really is.
This research is just one piece of the puzzle.
It's all connected.
Scientists all over the world are using computer modeling to study so many different things.
Absolutely everything from protein folding
To drug interactions.
to the evolution of viruses.
We're really entering a new era of biological understanding.
I agree. And our ability to manipulate these systems is growing so rapidly.
It's exciting, but also a little bit daunting.
I know there's a lot of responsibility that comes with this knowledge.
You need to use it wisely.
For sure, but I'm optimistic.
Me too.
I think this new understanding of biology is going to lead to some incredible advances.
For sure, in medicine and human health.
Absolutely. Who knows what diseases we'll be able to cure?
What lifespans we might be able to extend.
What mysteries of life will finally unlock?
The potential is limitless.
I love that.
Well, this deep dive has left me feeling incredibly optimistic
Me too.
about the future of science.
It's amazing how a topic like the MNS blood group system
Right, which seems pretty niche.
can lead to such profound insights.
About the future of medicine and human health.
It's amazing how every discovery, no matter how small,
Can have such a huge impact.
that's the beauty of science.
So much to think about.
We've only just scratched the surface.
Is there a final thought or a question you want to leave our listeners with?
Here's something to ponder.
All right, I'm listening.
We've talked about computer models as tools for understanding existing biological systems.
Right.
But what if we could use them to design completely new biological systems?
Ones that don't even exist in nature.
What would that look like?
That is a really thought-provoking question.
I know, it's a lot to think about.
We might need a whole other deep dive to explore that one.
We probably would.
But for now, I want to thank you for joining us
It's been a pleasure.
on this incredible journey into the world of blood groups and beyond.
So much to discover out there.
For those of you who want to learn more, be sure to check out the show notes.
We'll have links to additional resources.
And remember,
This is just the beginning.
keep asking questions
Keep learning.
and keep diving deep into that vast ocean of knowledge that's out there.
So much to explore.
Until next time, stay curious.